So we're welcoming Caroline Van Hemert and Matthew Smith with the USGS in Anchorage at the Alaska Science Center. And so with this multi-year die-off, a lot of the communities here, these are the birds of choice for food, especially the murres and the puffins. And so there's been a big food security interest in this. And tonight, can't wait to hear, so I'll be quiet and I guess Caroline's going to lead off. Okay, with that, I'm going to ask the speakers, ask your questions, feel free to use the chat, which is a feature at the very bottom. There's a bar with a toolbar, and if you go to the chat and click on that, you'll get a window that pops up and you can type in sort of like texting your question. We're going to hold off questions until the end. And also, I guess Caroline was interested to know, has anyone that's on this call right now, we know of at least one, do they do the coast surveys or have they turned in any of the birds? Anyone on here? All right, and I was saying to Caroline, one of the problems right now with the digital world we're finding ourselves in is we have a very big digital divide with the Bering Strait region, whereas here in Nome in the hub, we are internet connected. And unfortunately, in our more remote communities where a lot of this is ongoing, they do not have the bandwidth to do to actually connect and enjoy this presentation. So we are recording it. And of course, we have the Nome Nugget here and KNOM radio. And so they will be able to disseminate this information back out throughout the region. And we're really grateful everyone's on board here. If all the audience can keep their videos off, I'm going to click off. And that way we'll give Caroline and Matthew the benefit of all our broadband strength. Like I know how it works, I don't. So anyway, with that,
right.
Take it away, Caroline. Thank you so much.
Great, thanks. I'm gonna share my screen and just 1 second here. Okay, so I'm going to assume this is working unless I hear from somebody otherwise. Yeah, thanks so much for, okay, great. Thanks so much for having us, and thanks for everyone calling in. I know there's debates and other high competition activities happening, so thanks for deciding to join us instead. And as Gay mentioned, Matt, who is going to be co-presenting with me, and I both work at the USGS Alaska Science Center. My background is in avian ecology, so I study birds and a variety of wildlife and bird disease and health related issues. So tonight we're going to be talking specifically about Alaska seabirds and harmful algal blooms, and hopefully providing information that is especially relevant to the Bering Strait region. So in good government fashion, I have to give a little disclaimer, of course, that this information is preliminary, and it's kind of our way of being able to share the data that we've collected in a timely fashion. So we have a pretty intense data review process, which is just to ensure that all of the published information that comes out is as carefully vetted as it possibly can be. So by doing presentations and giving more timely information, we're able to hopefully go a little bit faster than the sometimes snail's pace that seems to result from these various review processes. So hopefully, don't hold it against me, but hopefully all this timely information is useful to people. So this is a nice follow-up. I think the last Strait Science, or one of the last Strait Science presentations was by Rob Kaler and Kathy Kulitz, if I remember correctly, and they provided a lot of information about some of the recent seabird die-off events in Alaska. So I'm not going to talk about all of these in great depth, but just to provide a little bit of background as to why we're studying this topic in the first place. So a lot of this really kicked off in 2015 and 2016 with a huge die-off of common murres throughout the Gulf of Alaska. And this photo here on my left, anyway, I'm not sure if that's how it presents on your screen, of just this line of mer carcasses was really the scene throughout Prince William Sound and so many regions in the Gulf of Alaska. And many hundreds of thousands of birds were found dead or estimated dead. And then shortly after that, in the Pribilof Islands, there was a die-off of tufted puffins. And I think there was just actually a paper that came out not too long ago about that. And then really relevant for the Bering Strait region, and some of you may have been involved in documenting this or even collecting samples, northern fulmars, short-tailed shearwaters, and a few other species in the Chukchi and Bering region in 2017. That die-off was fairly notable in scale, and I'll be talking a little bit more about that tonight. In 2019, there was a large die-off of short-tailed shearwaters throughout a variety of locations, mostly in western Alaska. And then 2018 and through 2020, multiple species, generally of smaller magnitude, so there hasn't been quite as much activity 2018 and 2020, at least so far. Concurrent with these seabird die-off events have been these really pronounced marine heat waves. So I don't need to tell anyone in the Bering Strait region that the ocean is getting warmer, but there's plenty of data out there to support these patterns. And there have been some really extreme anomalies, especially in 2015, 2016. But just in general, this rapid warming that is obviously contributing to some pretty major changes in the marine ecosystems. And of course, when you have broad-scale ecosystem changes, it brings up all sorts of new concerns. So everything from, what are these new fish that we're seeing in our backyard that weren't present here 5 or 10 or 20 years ago? Why are birds and other marine critters starving when it appears that there's plenty of fish, but maybe not the right types of fish that they are accustomed to eating? And then other questions too. This is an article showing the kind of graphic with the crab and the birds and walrus and whale and mussels. The title, if you can't see it very well on your screen, says "Deadly Algae Are Creeping Northward," and this was published in the Atlantic. And arguably it's a bit of a sensational headline, but it does bring up this topic that is increasingly of concern and of interest to people, is are algal species that can cause harm to people and to wildlife potentially becoming more of a problem in the north. Before we get too far ahead of ourselves, I'll just introduce this very basic concept of, what are harmful algal blooms? What are these HABs that we hear people talking about all of the time? And I know some of you are very familiar with them and perhaps have been studying them for a number of years. And then for other people, this is a brand new term. But in its kind of simplest form, it's by definition algal growth or the growth of algae that can injure or kill fish, wildlife, and humans. And there are a variety of mechanisms that can cause harm to people or to wildlife or fish. But one of the most common mechanisms is through the production of algal toxins. So the algae themselves actually produce toxins that then people or wildlife or whatever consume, and that can make them sick. And that's the part that I'll be focusing on today, is the production of algal toxins. And I should also mention that these harmful algal blooms are naturally occurring, but there are certain human-caused or anthropogenic factors, including warming, that can cause an increase in severity or frequency of these harmful algal blooms. So I think with research, it's always really important that we take a big step back and circle back around to this question of, so why should we care about whatever topic it is that we're researching? And since we're talking tonight about HABs, what is it about HABs that matters, and what matters especially now? And one of those that is kind of near and dear to my heart, because it's what my position is, my job is essentially focusing on, which is wildlife health. And we know that these harmful algal blooms can be really detrimental to wildlife health. One of the most well-known examples, and perhaps what really generated initially a lot of interest in harmful algal blooms in the context of marine mammals, is in California sea lions along the Pacific coast. So this image that's on the top left of my screen of a California sea lion is it's having a seizure. So it's been exposed to domoic acid and is having pretty severe neurological distress as a result. So we know that animals, and in this case sea lions, can get very sick and die from exposure to harmful algal blooms. In all of Alaska, but notably in Bering Strait region, since that's what we're talking about tonight, human health and food security and safety are really critical issues. And it's of course important to know if a food source is changing in abundance, but it's also more and more a question for people: is my food available, but also is it safe? And so the presence of algal toxins in potential food sources is something that is of growing interest to people in communities, as well as to anyone who's involved in human health. Impacts on livelihoods, so any kind of economic impacts, can be really quite huge and severe if there is a shutdown to a fishery as a result of harmful algal blooms. And this has been the case in Alaska to a certain extent, but certainly in other parts of North America and other parts of the world, it has had really pretty devastating effects on some shellfish industries. And then finally, I hope this Zoom thing isn't covering up my graphics, but oh, here we go, hide. There we go. Now you can hopefully see the whole screen. The other thing that makes this a topic that is especially important right now is that changing environmental conditions have potentially major impacts on the frequency and the severity of harmful algal blooms. And we all know that it's getting warmer, and it's getting warmer in northern regions more rapidly than in other parts of the world. And these changing environmental conditions may have pretty important implications for how much harmful algal blooms are an issue in our part of the world. And so just to bring this point a little bit more local, at least to Alaska, that when we think about HABs in a changing climate, there's a prediction that HABs will increase in both frequency and severity with climate change. And this is true for coastal ecosystems around the world, but especially in northern regions. And in Alaska, we know that at least in some cases, saxitoxin, which is one of the toxins I'll be discussing a little bit more in depth tonight, is positively correlated with water temperature. So this is a graphic, and you don't need to worry too much about what's on the axes, except to know that on the x-axis along the bottom there, that's temperature. And on the y-axis, so the one going vertically, is alexandrium, which are a type of algae that produce saxitoxin. That you can see there's a pretty clear relationship that as the water temperature is warmer, there's more alexandrium in the water. And this is a publication by Vandersea et al. In Kachemak Bay, so further south from Bering Straits, but still, I think, relevant for thinking about how warming may directly influence the presence of harmful algal blooms. And there are a lot of other factors, and many we don't fully understand in terms of what's producing these blooms, but we know that water temperature is a very important one. So I'm going to give just a little quick primer on two algal toxins that are produced by these HABs. And these are the ones that we're testing for currently in seabirds and in some of the things that they eat. The first one is saxitoxin, which you'll hear me talk about a lot more tonight because it's been found more frequently in our samples. And I'll either refer to it as saxitoxin, or you'll see this STX, which is just a abbreviation. And it's produced by a dinoflagellate called alexandrium. And it causes what's more well known as paralytic shellfish poisoning, or PSP. And there has been regular historic occurrence of saxitoxin and some fatality events associated with PSP, including, unfortunately, one just this last summer. Domoic acid is produced by a diatom called Pseudonychia, and it causes what's known as amnesic shellfish poisoning, and that's what you saw in that sea lion that was having a seizure. And although domoic acid we know is present in Alaska, to my knowledge, there has not been any documented illness or death. And I should also point out that the two toxins have very different mechanisms. So saxitoxin is a paralytic toxin, and so it typically causes respiratory failure because essentially there's paralysis of the respiratory organs. Whereas domoic acid is an excitotoxin, and that's why you see things like seizures and neurologic distress. One thing that we've found so far is that there may be some overlap between, at least clinically, what we see with birds with saxitoxin and domoic acid. So it may not be completely clear-cut in the field in terms of distinguishing the two, but there are some characteristic signs to look out for. So as I'd mentioned, that saxitoxin has been observed historically in Alaska, and plenty of observations more recently, including, this is from Southeast Alaska, some values that far exceed what we know to be kind of what's more or less safe for human consumption. And this is a value I'll refer back to if you look at that, where the red arrow is just to the right of that. The human regulatory limit for shellfish is 80 micrograms per 100 grams. And this is only important to know relative to other values. So that red line along the graph there is showing where the cutoff is for what we deem to be safe for human health purposes. And you can see that these are samples that were collected near Haines, Alaska, and this data is from Bruce Wright, the Aleutian Pribilof Islands Association. You can see that at times that those values are not only exceeded, but exceeded by an awful lot. And so values that would be extremely harmful, deadly, if you consume just a small amount. So we know that in southern Alaska, that saxitoxin is present, and sometimes present at very, very high levels. And so finding it in Alaska is itself not surprising. Maybe what changed our understanding a little bit was some work done by Kathy LaFay and others that was published in 2016, looking at marine mammals throughout Alaska, everywhere from Southeast Alaska to the Aleutian Islands to the Bering, Chukchi, and all the way up to the Beaufort, and a whole suite of different species of marine mammals, from sea otters to bearded seals to sea lions to harbor porpoises to belugas and humpbacks. And what they found was that saxitoxin and domoic acid were present almost everywhere they looked. And these toxins were present in both what appeared to be healthy animals, so hunter-harvested animals, as well as beached or stranded animals. And in most cases, these values were really quite low. But the big caveat here is that we really don't know the sensitivity of wildlife species to these toxins. But so we don't know, did the toxins in some of these cases make the animals sick or die? But we do now know that saxitoxin and domoic acid are quite widespread throughout Alaska. And so that brings up this question of, well, is it harmful, and is this something that's changing, or has it been present for a long time? When some of these seabird die-off events were happening, of course, one of the questions was, well, could algal toxins have been involved, or what might have killed these birds? And at the time, when we were trying to think about how we might approach this question, there wasn't a lot known about algal toxins in seabirds. We know that, like marine mammals and any other marine consumers, that they're exposed to toxins through the food webs, through what they're eating in the marine environment. There's surprisingly little information about the direct effects of either saxitoxin or domoic acid on birds. But there have been a number of cases of bird die-offs that were pretty definitively linked to saxitoxin or domoic acid exposure. And here's just some examples of seabird deaths that have been attributed to harmful algal blooms over the years. And there was one case actually in Alaska just a few years ago among Kitlitz's murrelet nestlings, where these chicks were being fed forage fish and pretty quickly became ill and died. And then subsequently they were able to test the tissues and some of the fish that were in the nestlings had consumed, and found fairly high levels of saxitoxin. But it can be really hard to directly link cause and effect for something that we don't have a lot of background information about. And we had started off some of our seabird work in HABs with the MER die-off of 2015, 2016, but we quickly had an opportunity to do more research that's pertinent to the Bering Strait region. associated with that die-off that I'd mentioned earlier that primarily affected northern fulmars and short-tailed shearwaters. And there were about 1,700 birds that were found dead, the carcasses that were actually counted and observed. And that might not sound like a ton, but that almost certainly represents just a small fraction of the total number of birds that were involved in that mortality event. And I should just put a big shout out now to community members and those who participate in the COAST program and others who have been involved in data collection, both carcass counts and carcasses themselves being provided for research purposes so that we can try to understand what's going on with these birds. And it's so critical that we not only are able to receive the samples, but also to have local eyes and ears and people's observations so that this information can even come to people's attention to be able to understand changes, because obviously it's local folks who really have the most background and ability to be able to see things, see changes in real time. So thanks for everyone who's been involved in that effort and continues to be. Our sample collection associated with that die-off were not huge numbers, but enough to give us some idea of what was going on. So we had northern fulmars, 16 of those, a couple of common murres, a few short-tailed shearwaters, and then just a single storm petrel and tufted puffin. And again, this was really community-based carcass sampling. And the typical stream for us is samples come in from various places. They often go to Gay or wherever the regional hub may be for a particular area. And then Rob Kaler and others at Fish and Wildlife have really spearheaded the effort to make sure those then get to the people who can do the necropsies and collect samples and try to make sure that all the birds are being treated in the same way so that we can collect as much information from each of them as possible. And then we receive those samples and are able to test them for saxitoxin and domoic acid. And that was the same for this particular die-off event. And so what did we find for this year? We were fairly surprised, actually, to have detected saxitoxin in almost 90% of the northern fulmars, which was the species that was by far the most commonly observed in this die-off. And in some cases saw values that were high enough to really raise some eyebrows. The maximum value was about 63 micrograms per 100 gram. And then you can see there's a variety of other concentrations in different individuals. And remember, this isn't a huge sample size. I think there's 15 birds, but enough to make us start to suspect that there could be something going on in terms of exposure to saxitoxin. The pretty consistent finding across all of these die-off events is that birds usually come in pretty emaciated, so they haven't fed in a while. And as Gay mentioned in her introduction, we don't know why that is. We don't know if it's because there's not enough food available or the right type of food available, or if they're somehow compromised and not able to forage as they normally would. And I think it's going to take us a little while to answer that question, but we do have some additional work lined up that Matt will talk about that will hopefully help us get closer to that point. So I can't answer that from these values. They're just what we observed in the carcasses of birds that were found dead. You'll notice that the highest value is in stomach or intestine or cloaca, and that's to be expected in the gastrointestinal tract, that those are values or those are parts of the sample that were most recently consumed. Saxitoxin and domoic acid are excreted really quickly, so they go in and out of birds very rapidly, which Matt will also talk about a little bit from our captive work. And that can make it a real challenge to have good real-time information that tells you how much a bird actually consumed over a recent period of time. And I thought people might be interested in seeing locally where these values were being observed. Oh, and I forgot to mention, I'm just going to go back for a second, that it's very important to keep in mind that although our highest value 63 was well in line with other die-off events that have been associated with saxitoxin, we don't know what actually killed these birds, and we can't say definitively that it was saxitoxin. And even more importantly is to keep in mind that the human regulatory limit is still above that. So that's maybe some of the good news here is that, you know, saxitoxin was present in these birds, but in none of our samples did they exceed that human regulatory limit of 80 micrograms per 100 gram. And we're not in a position to advise on human health, but it does give us some indication that these values are not, you know, astronomically high, at least what we've seen so far. And then just looking a little bit more specifically at where these samples were collected and what the values look like in different regions for people who might be interested or may even have actually collected some of these samples. I'll let you look at them individually, but just to help you interpret what this looks like, the number of birds or the number of samples we had, and then the red ones are birds that were positive, and the white ones were birds in which saxitoxin was not detected. And I should just emphasize again that this is just for saxitoxin. We didn't have any domoic acid detections from that year at all. And you can see that in some cases there were just trace values, and then some of our highest values were from the Pribilof Islands. That 63 micrograms per 100 gram was off of St. George, and some as well up in Shishmaref and Gamble regions. And Unalakleet, we didn't have a positive, but it was just one bird. So these are small sample sizes, so take them with a grain of salt, but enough to start giving us a little bit of a picture of what might be present for the birds to consume. And then I'm going to actually turn it over to Matt now to talk about some of our additional results from 2018 and beyond. So I will stop sharing my screen, assuming I can figure out how. Stop share. Yeah, thanks a lot, and we'll answer questions more.
Okay, so I will try to start sharing my screen here. And if Gay or Caroline wants to let me know when it comes up here, if it's working.
It's working.
Working?
Yep, you just need to hit the presentation. I think the little screen at the bottom, or however you want to do it. There you go, you're in.
How's that look?
Brilliant.
Okay. All right, well, thanks, Caroline, for taking over the introductory part. Like Gay said at the beginning, my name's Matt Smith, and I work with Caroline at the Alaska Science Center here in Anchorage. And I'm going to try to just keep moving on to where Caroline left off. So if I can get this screen out of the way. So following the 2017 die-off and the elevated saxitoxin levels we found in fulmars in the Bering Strait region, there started to be a little more interest, at least on our ends of things. I'm sure there's been concern about this for some time now about harmful algal blooms in this area. And in 2018, there was an additional seabird die-off of moderate scale throughout this region. It looks like, based on the available data, that it was roughly the same size, maybe a little less. Some other folks might know better in size than that of 2017. And again, you'll see a recurring theme through all this, that the carcasses and count information were largely collected by community members, and the data was compiled by the folks at Coast. And so as far as sample collection in 2018, we got multiple species: kittiwakes, thick-billed murres, horned puffins, some of the same species you'll see throughout the 3 years here that were collected during these die-offs. We did get one greater scaup that someone collected around the Norton Sound region this year. And this was a pretty small sample size. We received 14 whole individual carcasses from the summer and fall of 2018. And that came back from the National Wildlife Health Center as 58 tissue samples from 6 different species here. And so this was the smallest sample size of the 3 years that we've done testing from this region. And none of the samples that we analyzed had any quantifiable levels of toxin. You'll hear me say quantifiable levels, or there's a difference between quantifiable and detectable. And I can explain the difference between that at the end of the talk if anybody has any questions. We did detect one trace sample of saxitoxin in that greater scaup that came from Norton Sound. And so the 2017 and 2018 results have already been released, and I think we briefly went over those when we were in Nome last year and gave a presentation. So the 2019 samples here that I'm going to talk about now these are all new data and haven't been released. So I think this is what a lot of folks are waiting for. And the 2019 die-off was a significantly larger event. It ranged from the Aleutians in Bristol Bay all the way up past Point Hope through the Bering and Chukchi region. And this primarily was comprised of primarily short-tailed shearwaters. And again, this was all largely pushed through by community members collecting carcasses that they found on the beach. And this was the largest sample size we've had out of the 3 years. And these carcasses were received from 9 different communities, and 60 individual birds were submitted through, typically, I think, the U.S. Fish and Wildlife Service, Rob Kaler here in Anchorage, and then on to the National Wildlife Health Center, who then sends the samples back up to us here in Anchorage to analyze. We ended up analyzing 203 tissues from these individuals for 2019. And the sample collection for this die-off was a little more diverse. Like I said, the largest sample size out of the 3 years. Similar species, a few crested auklets in here. And then we also had some other species that we don't normally get. One Pacific loon that I think Gay sent in, a couple pectoral sandpipers, pelagic cormorants, and eiders. And now most of these samples did not contain any detectable saxitoxin or domoic acid. There were several individuals total that did have quantifiable levels of toxin, 3 for domoic acid and 4 for saxitoxin. One of these individuals had toxin detected in multiple tissues, and you can see here on this chart, there was a different mix of different species. Several were collected in Shishmaref, others in Point Hope, and then one from Nome. And as far as toxin concentrations go, all of these toxins were detected in digestive tissue, either upper or lower GI samples or liver. And they were all lower, very low, or trace amounts. They were significantly lower than what we found in Fulmars in the 2017 die-off that Caroline covered earlier, and nowhere near the regulatory limits for what the FDA sets for human consumption. I think the highest detection we had was 2.7 micrograms in the lower GI tract, and I'll come back to detecting these toxins in the GI tract and how that ties into some of the experimental work we've done. And just for some preliminary results, we were able to get samples from the 2020 Seabird die-off event. This was a overall lower magnitude die-off from what it looks like. And not nearly as many samples, just slightly more than 2018, but similar species, common and thick-billed murres, some eiders, short-tailed shearwaters, and one black brant that I believe was hunter harvested and submitted. And as far as these results go, testing in our lab has been largely delayed due to the COVID-19 pandemic, and we've been working from home since March and only recently got approval for part-time lab work. But I was able to get all the samples sent up from the National Wildlife Health Center in September, and we're currently trying to work our way through the sample backlog. We were able to test 5 individual birds screened so far, and there haven't been any detections of either the toxins from those 5 individuals. So to quickly summarize what we found in this region, mortality events have been responsible for thousands of seabird deaths between 2017 and 2020. With the exception of the Fulmars in 2017, there have been few detections of algal toxins in any of these birds. And similar to other work that we've done in different regions, we still don't know much about the effects these toxins are having on seabirds. Testing opportunistically collected carcasses is a really great way to determine if HABs are occurring in the region. And it's great that we're able to have these community members involved to send these samples through and then get these data back released to the communities. But it's also difficult to understand the actual physiological effects these toxins are having on these birds after they're ingested. So we have done some experimental work in an effort to answer some of these questions that are really hard to get at through opportunistic sampling. If anyone, some folks may be familiar with this, or if people attended the presentation that Caroline gave in NOAA or at AMSS, this might be familiar since I've talked about it a couple times. In 2018, we teamed up with researchers at the National Wildlife Health Center that were conducting an experimental dosing trial on mallard ducks using saxitoxin. And the goals for this were to determine the lethal dose of saxitoxin on these birds, since there's been very little done for birds. Most of it's done with mammals. And also to determine the tissue toxicity and tissue routing pathways to see where this toxin goes after the birds ingest it. So just to give you an overview of the main takeaways from this study, similar to what we found in seabirds from the Bering region and other places we've sampled birds from the wild, most of the saxitoxin has been confined to the GI tissue. You can see here the differences on the top between kidney, muscle, liver, and lung tissue. All the levels were low or very low. And then large intestine, small intestine, upper GI, they were all high or very high. And there were no toxins detected in heart, blood, or brain tissue. And these are all for birds that died acutely after dosing. For birds that survived the lower doses and lived through this full 7 days of this study, we didn't detect any quantifiable amounts of toxin in their tissue, which to us may indicate that within a week of ingesting these toxins, the majority of it may be out of their system. And Bob and his group also looked at saxitoxin levels in fecal samples of the birds that survived. And this figure here shows the results from the time immediately after dosing at 0 here on the left of the figure, out to 168 hours or 7 days. You can see this large spike at the beginning here, followed by steep declines, and then these peaks and valleys over time here, it's slowly declining. And the main takeaway from here, and what was really interesting to us, is that we didn't detect any quantifiable toxin after 48 hours in any of the fecal samples. We didn't detect toxin in any blood samples at any time. And this just shows that we may have a really short window between when these birds ingest the toxin, if they don't die acutely, to be able to detect it through these less invasive methods in the wild. So it can be a really short time period for when we can find if these birds were exposed to toxins or not. So just to quickly wrap up what we found for as far as HABs in the Bering region, they are present in seabirds in this region as well throughout Alaska. The highest levels appear to be in the stomach, intestine, and GI tissue. And the die-off in 2017, particularly in fulmars, may have been linked to saxitoxin exposure. Other years, we found very few detections, pretty much just a couple from the samples in 2019, and none of them were high enough to attribute to a saxitoxicosis. And there are also maybe other confounding factors, food availability, ecosystem shifts. It's really hard to apply causation of these toxins to seabird die-offs. And there are many unanswered questions about the impacts of these HABs on seabirds. It seems like every time we do a new study with this, it actually raises more questions than it answers. As far as future work is concerned, there are still many unanswered questions, many missing pieces of the puzzle. Some of the current projects that were supposed to be worked on this year that got delayed due to the pandemic are looking at the distribution of HABs. Some of the folks from the Seabird group at the Science Center that we collaborate with are going to look at where are HABs occurring, what species are being affected, and then also how do these toxins move through the food web. Algal toxins are very much a bottom-up effect. They're produced by really low-level producers, phytoplankton, and then they move up through the food web. There's not a lot of information on how long that takes. So that's one big project that we're hoping to move forward. And then we're also building off of the mallard study, hoping to do some additional captive trials. We're collaborating with the Alaska Science Center to do a project with MERS and look at sublethal effects of toxins. So if these birds are ingesting toxin in the wild, but it's not enough to cause acute mortality. Can there be detrimental effects by just, for example, affecting their foraging ability? So they may not actually have to be poisoned to die, is some of the questions that we're hoping to look at. And if folks have any other questions, we do have a webpage up through the Alaska Science Center website where you can find more information on this topic. There's some pages for some frequently asked questions, as well as just some information. We're going to try to keep more up to date, since it sometimes does take a long time for us to get these results out to folks. So you can follow this link here and get in contact with a lot of this information. And with that, just like Caroline said, I want to reiterate that a lot of this is largely only made possible by community members collecting these samples and then submitting them to folks that can then get the samples back to us to test. So this would be really hard to do without those people doing that. So a big thank you to people who are collecting all these things, as well as everybody else who helped us out with this project. And I think with that, we can answer questions if anybody has any. If I can figure out how to stop sharing.
All right, you go to the bottom of the screen. There you go. All right, well, thank you very much, Caroline and Matthew. Very much appreciated. I don't know how we clap in Zoom, but that would be a good solid round of applause. And there we go. And I'm sure people have questions. I know I do, but let's other people go first. Anybody have a question for Caroline or for Matthew? All right, while everyone's thinking of their question, I'm going to ask mine then. I guess I'll jump right in there. So for the communities in the Bering Strait region, we had very many, over that course, those years, we had many birds dead on the beach that were reported, and we had many more that were turned in than your sample sizes may show. Your sample sizes are kind of low. Is that because of the condition of the birds, like they were not pristine enough, or were you looking for certain organs, or were you not able to get your hands on every single bird, or is it the cost of doing analysis? There seems to be a discrepancy between the number of birds turned in and then the sample sizes, like 2018 especially, I think, maybe.
I think whatever we were able to get, we analyzed. So it wasn't a cost limitation thing. Sample volume can be an issue. So, I think probably some people are aware of the limitation with when birds are collected and they're starving, there's going to be very little tissue from gastrointestinal tracts. So that, is one limitation. And then kind of depending on, if they're really degraded, then that's something where hopefully wasn't listed on our to be done list, but we hope to do some work on looking at degradation over time, so that like if you pick up a bird that's pretty well aged on the beach, are you going to be able to detect toxin at all, or are the microbial processes or anything else affecting that? So we don't know the answer to that, but typically the Wildlife Health Center is limited on what they're willing to take for necropsy, because if it's too far along, they can't say much of anything. And so, we haven't so far been testing things that are really rotten, just because there has been some work done up to this point suggesting that might compromise results. But I think everything we've received, we have tested up to this point, besides 2020. So I'm not sure if there's other samples that didn't get included, or
Well, I think the National Wildlife Health Center was archiving birds too, so there may be more in there. I know they would get a bolus, a collection, and of maybe 8 to 10 birds sent to them, and then they might analyze 3, and then they'd put some in the freezer for a later date. So you might want to ask if you want more samples. They may actually have some. Don't know.
We've definitely gotten samples additional to those that they've necropsied. So like even for 2017, I don't know if you remember, but in writing up those results, there was a subset that were necropsied, and then we've requested that we get, and I think at this point that's a pretty standard thing for them to do. They will subsample even if they don't do a full necropsy. And so yeah, basically whatever we're getting, we're testing. So hopefully if you see something really anomalous, then let us know. But I think most of them should have been accounted for.
Okay, thank you very much. That just caught my eye. Thank you, because it really interesting to me. I think so. It looks more that it was just plain starvation, which has, and not HAB related.
Yeah, I would say that's probably the best hypothesis at this point, that there are so many underlying changes that I would not be quick to point my finger at HABs. But as Matt mentioned, some of the sublethal effects I think are really interesting. And we know from field collected samples that it's going to be very difficult to be able to pinpoint a HAB related die-off unless you have a lot of birds that died acutely and very, very high levels. And so if birds are being exposed to something and they're somewhat compromised, that could be a contributing factor. So that's one of the main questions, because there's been so much focus on HABs, is, how big of an issue is this for seabirds? Should we be concerned, or should we focus our efforts elsewhere on trying to understand population dynamics and other things that are contributing? So yeah, by no means are HABs the only change that might be affecting seabirds. And this is just we focus on this piece of it because that's what the talk is about. But there's other folks with Fish, Wildlife, and USGS doing a fair amount of other food web related work too. So
great, thank you so much. I mean, that's a big question. Is it the environment? Is it the ecosystem shift in the northern Bering Sea, or was it the warming, emerging issues of HAB? So thank you. Anyone else have questions for Matthew or Caroline?
Yes, I do. This is Rafaela from Barrow, Alaska. My first question is, so, any thoughts, because in 2017 you said you only detected STX but no domoic acid, but then in the subsequent year there was domoic acid detected as well, right? Am I right about that?
Yeah, they were very, very low. I should have mentioned that in the slide that had the levels. In 2019, we had 3 individuals that we detected domoic acid, but those levels were flirting with the edge of not detectable. They weren't quantifiable. They were very, very low. So nothing that would stand out and that's what we found in other samples from some of our other work too, is just very low trace levels of domoic acid in some of these samples.
Okay, thank you. And then I have a somewhat gory question, and I apologize for everybody on the call, but I do have to ask it. Since this was a lethal, dose or toxicity study, and I'm sure you monitored the clinical progression to death in these animals, as part of your endpoint. So I guess my question is, will that information be coming forth soon? I mean, will you guys truly, I guess my question is, how much monitoring besides, observational study you did during the process of dying? So the reason why I'm asking, of course, is, are there any clues that we can get from it, right? From observing an animal and say, just like for other diseases or for other toxic events, you have some, characteristic signs. I assume it was just, cardiovascular, drop out. But it would be interesting to know, some question, are you guys going to publish that? Is that going to be part of it besides just telling us that dose and then we were detecting? But is there actually a clinical correlate? Was that clear?
Yeah, as far as like reactions to the ingestion of saxitoxin on the mallards, is that what you're asking?
Yeah.
Yeah, and that is going into the, Bob, who's the lead author on that project, just submitted the manuscript that does. They did record all the observations, and then they also performed necropsies on all the birds, whether if they died acutely or they were euthanized at the end of the study, and they did not find any clinical signs, any physical signs, lesions or anything.
No histological signs, right? But the clinical stuff was overlapping with some of the domoic acid things that have been reported before, right, Matt? Like there was
Okay.
seizures, vomiting, diarrhea, like a lot of gastrointestinal distress. But then the seizures, I think, were one of the things that is often reported for domoic acid, but that was seen with the mallards too. And then Matt, can you remind all of us what it was when birds died? It was typically quite rapidly.
Yeah, and
Like it's
the really obvious clinical signs came on really fast and then were immediately preceding death. So they didn't go through seizures for days where you could, basically if a bird ever started convulsing, it died within minutes afterwards. Some of the other signs, and there was one other study that looked at domoic acid in MERS, and they found similar thing as wing drooping. They can't, when the birds are sitting or trying to raft on water, they can't fold their wings back along their back like they normally do. Their wings droop to the side, and head shaking, tail shaking, excessive shaking was another one. But once birds started to move towards the convulsion method, they weren't really sure whether that was a true seizure or if it was more cardiovascular system, lung paralysis, where the birds are, on the way out and then went through violent convulsions right before the end. But that is all described.
Okay. All right. Can I ask one more question? And thanks for both of you answering, Caroline and God damn it, forgot your name again, Marcus. Sorry about that. Didn't want to swear. So from the time point of when you dose the birds to actual, deaths, are we talking 10, 15 minutes? Is that about it?
Yeah, for some.
I'm just trying to.
Yeah, within 15 minutes. I think the longest
Okay.
for the longest bird that the longest time when a bird still died acutely was still under an hour. So none of it was, or maybe just over an hour. I'd have to look back. But it was all really fast. Some of the beginning of the up-down, LD50 trial was, I think, 11 minutes, 12 minutes, and it slowly went up from there. But they all died within an hour, and some really quickly.
Yeah. Well, so thanks for sharing that. And I think, that kind of information, although it is gory, will be really valuable, for the hunters and for the communities, because there's a lot of observations, actually on animals that are foraging, right? And so if you are observing deaths within that short period of time, also with some of the signs that you're describing, that's going to be really something for, for hunters and for the other experts to hook into and be able to say, hey, we got to get that bird because that ain't, that's actually telling us that we may have a toxin involved. So I think it's going to be very good if this information is published, but I would really suggest that maybe at some point when you guys are closer to make a, more community friendly, presentation on that, where we did that once for the walrus community, where we took what we knew from other animals, right, and talked to hunters about what the potential, what they could be seeing and how it presents itself in other animals. And, that was, well taken up, and people really were able to use it to say, okay, so now we know what we're looking for. And I think we have, some thoughts about this. But your study, I guess, is, and the subsequent study is going to be the first ones where the animal, as dramatic it is for those animals, will actually teach us something for the future, how to look out for it, So thanks. So I think that would be very valuable, Caroline and Marcus, if you guys would do that.
Yeah, and hopefully
drill it down to.
Oh, go ahead, Matt.
Yeah, there was a lot of interest. I've given a couple talks on the results of that study now, and that's one of the questions that keeps coming up, and that's what we're hoping to get at in that study at the Sea Life Center. We're moving more specifically to seabirds, and then rather than do an LD50 study where each bird is going to die, we're going to do more of a, because Bob was able to determine, like, okay, we know what levels these birds need to run into the environment to die, but what might happen if they're just either exposed to lower levels over time, and what's their reaction going to be like? So what might people see if birds aren't dying acutely? Because that's going to be pretty obvious when if a bunch of birds die in one specific region all of a sudden. But if they're just ingesting this lower level of toxin over time, and it's keeping them from foraging or causing them not to be able to raft properly while they're out in open water, that kind of stuff, like what that might look like. So I'm hoping to figure out with MERS at the Sea Life Center in the next year or so.
So I have one more question, and that's, you know, whoever wants to answer this one. So, you know, this incredibly, you know, rapid clearance of your 48 hours. You know, so we did a study in, you know, in bowheads and in some other, and seals, you know, these were harvested animals to look at, you know, comparison between stomach contents, you know, feces, and then actually that's Cassie Lefebvre's work of looking at, you know, if prolonged storage of samples is going to change the, you know, detectability, right? And for domoic acid, we pretty much can say it sticks around. If it's there, it will stick around. And we have a, you know, a sample even from a seal that was put in an ice cellar, right? A mummified seal that had been in there for 70 years. And we were able to, you know, detect the domoic acid. But it looks like for STX, that's not holding up. So do you have any thoughts on that?
Are you saying it's not holding up based on what you're finding with Kathy, or just another?
Yeah, it just sounds like that it's a, you know, I mean, unless I misinterpreted that, you know, one graph where, you know, you saw that rapid, that you, maybe
yeah,
if you
okay,
could go back to, do you remember which one I'm talking about?
Yeah. Okay, so yeah, just to clarify that it's rapid clearance from the bird's body, not rapid decline of the detectability in a sample. So I think once a sample is collected,
sorry.
Yeah, no, okay.
Sorry.
And it's
Okay.
if it's frozen, then we know it's in pretty good shape. What we don't know, and we'd like to find out more about, and Matt was like set up to start, and then we basically had to leave the lab, but is if a sample is sitting, you know, at room temperature, or even not room temperature, but whatever it, you know, might be 40 degrees for a period of time, would that change the detectability in a toxin? But that particular graphic was just the clearance from a bird. So basically, if a bird's consumed it and you're trying to detect it, you don't have very long before it's going to be gone. So
Thank you.
It wasn't in a sample, and that's one thing we're hoping to work on. And Kathy's done a little bit of that work recently that hasn't been published or released yet, but yeah, so that's because that's an important factor of if these birds are washing up on the beach and they've been there for a week and they did have toxin when they died, how much of it is still detectable by the time the sample gets all the way back to me. So
I want to just follow up one thing from the captive study that I thought of too, is that the mallards were dosed with oral gavage, whereas with the seabirds, we're hoping to mimic something across the board and, you know, more similar to what conditions they might experience in the wild. So actually dosing fish and then having them consume fish, and then doing some live foraging trials where hopefully they can be trained to swim after fish, and then seeing how their ability to do so might change with exposure to saxitoxin. So trying to get at more of those real world correlates. So hopefully, Raphael, we can answer some of those questions a little bit better before too long.
Well, great, thank you. Any other questions? And thank you, Rafaela. Any other questions for Caroline and Matthew Smith?
Yeah, this is Kate.
Hi, Kate.
Just to clarify something, so Pete and I do a coast beach here, and we've not turned in any of our finds because I was under the impression that they really had to be very fresh birds, and we don't find those birds that are that fresh. But do I gather that if it seems like there's a chance that the digestive system is still intact, that it's worth turning in, or do they really have to be very fresh?
So for that, I can say that what we've been told by the National Wildlife Health Center is the birds have to be intact. And I think they want to cut it off at that, because we can get a lot of predated birds. And I don't know if it has to be totally pristine, but it has to be intact so that they can get at there's so much they're looking at per bird, you know, doing the necropsies, that they'd rather have them intact than to try to tease out a little piece of intestine and go through it. I mean, they need that's what they've been telling us anyway. Carolyn and Matthew, do you have any other?
Yeah, so it's kind of it depends on what your question is, of course, with all of this. So for the necropsy to, you know, really get at what might have killed the birds, if you can't do decent histopathology, then you're really limited, and you do need, you know, relatively fresh, although it's all relative, of course, when you're dealing with remote locations. For the algal toxin work, you know, if it's not rotten, then it's within the realm of possibility as far as what we know so far. And in some cases, you know, the Wildlife Health Center may not be willing to necropsy all the birds, but I think the thing to keep in mind is if it's part of a larger event, then that, in my mind, elevates it a little bit over the really scattershot, like we expect a dead bird here or there. And so if that's the case, it may be a lot of effort on everybody's part to maybe get pretty limited information, and then in the end you can't say a whole lot about it anyway. But if you're seeing something anomalous that's occurring across a broader scale, I'd say more samples are typically, you know, better, because we can get a much bigger picture of what might be happening across that area. So it kind of depends, but it never hurts to reach out about samples. It's hard not knowing like the level of freshness or where your location is. If you're, you know, if getting something shipped requires 4 different flights, then that's obviously a different deal than if it's possible to get it quickly to Nome, and then can even hold on to them and ship them in a batch or something like that. But yeah, I would say, I mean, we are certainly willing to test additional samples, even if they're not in perfect condition. But if they're just, you know, these scattershot bird here or there, it probably isn't going to provide a whole lot of information unless we happen to get lucky with a high level detection or something.
Okay.
Does that help you, Kate? Sorry.
Yeah, it does. We're here in Nome, and
Okay.
are these die-off events? Yeah, I'll maybe be bringing you some, Gay.
All right, I've got a freezer in the office now, so it's all good. All right, any other questions? I don't mean to keep Caroline and Matthew on for a long time, but want to make sure everyone gets a chance. And I just want to reiterate, go ahead. I just want to reiterate how important those observations are and the behavior. So we really would be interested in helping disseminate that. When we had the avian cholera, there's some very distinct behaviors that the birds exhibit, and that was very helpful for the National Wildlife Health Center. Kind of help them help us to sort of make sure that what we were seeing was being reported, certain behaviors and things that the birds would do with avian cholera. And so if there's something that there are so many silent partners to your project that aren't on, you know, Zoom right now. And the level of expertise and behavior is really high throughout the Bering Strait region when it comes to seabirds. And so those kind of clues would be really helpful to give to people so that they can maybe help you help the region as far as getting samples and from specific behaviors that are seen and getting them to you quickly would be really helpful for both you and your studies, and for, of course, getting those results back if we have a problem. So
Certainly, yep. And also, I should just mention that people are more than welcome to reach out to us to that website link, which is just as easy to find by Googling USGS algal toxin seabirds, or going to the Alaska Science Center website. But there's our contact information there too, so
awesome.
You're welcome to contact us directly if that's helpful.
Yep, for anybody. All right. With that, I guess I guess we'll close it out.